Okay, so the last video that I just did covered the basic anatomy of a neuron. And so this is a typical neuron. We went over all the pieces and what comprises a specific neuron. So the next thing that is important that we cover is how the neuron functions at rest. So we're going to talk about how it functions at rest and how it functions when it's doing something. So if you look at a neuron that doesn't have any sort of stimulation, it's not doing its job, it's what we refer to as in a resting state. And so when we have a neuron that is resting, it has a potential, which we refer to as the resting potential. So it's ready to activate, it's ready to communicate, as long as it receives sufficient stimulation. If a particular neuron is sitting and waiting and doesn't get any stimulation, then it maintains its resting potential. So imagine that we have, and I'll draw this sort of right here real quick. Imagine that we have a neuron, and this particular neuron is right here. Okay, here's the cell body with the nucleus in it. We have the dendrites coming out, okay? And then we have an axon here, and then we have the terminal boutons down here. Okay, so this is a very, very crude neuron. And it's got its dendrites and it's receiving some stimulation. Let's say we'll call this another neuron up here. Okay, and this is the axon, and it's a really small neuron. But let's say that this right here is where we're getting some stimulation right here. Okay, and let's say there's another connection from another neuron and we get stimulation here. And let's say sometimes we actually have connections that aren't on dendrites but directly on cell bodies. And let's say it's getting some stimulation here. So it's getting some messages from all these other neurons and it's saying you need to do something, okay? It's like you need to activate. What happens is that until it receives enough messages, it's just gonna sit and not do anything. It's just going to sit and wait. And this is what we refer to as this idea of resting potential. This resting potential is the state where it's prepared to activate, it's prepared to communicate as long as it gets the message. But as of right now, it's not getting enough stimulation. So one thing that we have to think about here is this idea of what's inside of a cell, what's inside of a neuron. And so what we find is that all neurons contain ions or charged particles. And these ions are located inside a cell and they're located around the cell. And so we have positive ions and we have negative ions. So positive ions are cations and negative ions are anions. So here it says position. Obviously this should say positive. So I found that typo, sorry about that. So when you are talking about a particular potential, okay, we have to remember that it's inside the neuron and outside the neuron, we have these positively and negatively charged ions. So at resting state, we see that, and this is something that you're going to need to write down. So this is something that if you have your notes out, this is something that you're going to be tested on. Generally speaking, a cell is at negative 70 millivolts at resting stage, at resting potential. A cell is sitting at negative 70 millivolts, meaning that it is more negative on the inside than it is on the outside, okay? So this is going to be very important here in just a few minutes. So a cell on the inside is sitting at a negative 70 millivolts, okay? Now, so we have positive ions, which are cations, negative ions, which are anions. And the mixture of those charged particles will determine if we have a more positive or a more negative charge. And the inside of a neuron sits at a negative 70 millivolt charge. Now, this is the reason why it's sitting at a negative 70. So we have four major ions that are found in and around the cells. And so these aren't the only ones, but these are the major ones that we're going to talk about. So we have here, we have sodium, which is Na positive, okay, and chloride. Sodium and chloride are found outside of the cell, okay? So sodium and chloride, we see much more of that outside of the cell. Now, this makes sense if you think about it, because there is fluid that's in the brain. It surrounds the cells, it's in everything, and it's described as a very salty fluid. If you were to crack open a skull and lick a person's brain, it would taste kind of salty. Okay, I'm not suggesting you to do it, but it is certainly something that would taste kind of salty. Okay, so it's a salty solution concentration because we have more sodium and more chloride in that fluid. Okay, now, so here you can see that there's not as much sodium and chloride inside the cell. So the sodium is represented by the orange, and the chloride is represented as the blue. You can see there's a really high concentration of these positive ions or cations, okay, and a lower proportion of the negative ions. And inside the cell, we see that we have potassium and large proteins, and we have a much higher capacity concentration of potassium and large proteins that inside the cell than outside of the cell. So here we can see that we have a lot of these negative large proteins, and we have a smaller concentration of potassium inside of the cell. Notice when you look outside the cell, there's very little in terms of concentration. So because there are more negative or anion charged ions in the inside, then it's going to rest, the resting state is going to be more negative, okay? And outside of this cell is more positive. Now, so here we have a more concentration of potassium or positive ions and protein ions or negative ions inside the cell. We have more of these negative ions than anything that are sitting inside the cell. And so the cell is sitting at a much higher, much more negative concentration. Here we have more sodium, which are positive, and chloride, more negative cells. We have more of these sodium cells, so it's more positive on the outside. Think of this as the axon of the cell, and the axon, you can see there are more negative ions, and outside of the cell wall, we see more positive ions. So, very important here that while the cell is at resting state, it is sitting at that negative 70 millivolts. So, if it receives that stimulation, and it's enough, and it crosses the threshold, if it's enough to start a party, then what ends up happening is that that particular cell will then activate, okay? Now, it's only once this threshold is met that a cell can then activate. So we call this process of activation action potential. So when we see that enough stimulation has been produced, it will produce an action potential. We call this firing. The cell fires, okay? And so essentially what this is that those electrically charged particles or ions will actually move across the membrane, move across the axon membrane. And so it is this electrical impulse that begins at the cell body axon hillock where the cell body meets the axon. It begins there, and then it goes all the way down the axon to the end. And that's only if it meets enough stimulation. So it's sitting at a negative 70. And if it gets enough stimulation to change that charge, then we'll see an activation happen. So we're going to talk about that process. So here we see, this is probably one of the most basic tables, the charts that's used to show an action potential. This video is very, very good. If you are not sure about this whole thing and the action potential is something that you're not sure about, then I really suggest that you watch this video. okay? I think that everyone should, you can stop right now and you can watch, or you can wait to the end. I'm going to spend just a couple minutes on this, and then you can go ahead and go to the video and watch it so you're nice and in order. So let me explain this to you, how this works. So this would be sort of a graph of how it works, okay? So this is over the course of 3.5 milliseconds, okay? So we see 3.5 milliseconds here, and this is the electrical charge of the cell. So like I've said, it rests at -70. So if it's not getting any messages, if it's not getting any sort of stimulation, it's going to sit at -70. Now, let's say that it's at resting state, and then it gets a bump, it gets some sort of hit from another cell, okay? So another cell gives it a signal. Well, here you can see that if it gets a signal here, nothing's gonna happen, okay? It's not met the threshold. It's not until it meets, and this is arguable for some cells over other cells, about a -60 or so, between -60 and -50. Once it hits this threshold, once the threshold is hit for that cell, all is done. That cell is going to activate so it can get lots of different messages from other cells. Once the ions move across the surface and change the charge from -70 to the threshold, which in this case we'll say -60, okay? Once it has met that threshold, the cell takes over, okay? It needs no more stimulation from any other cells. It doesn't need any more communication. The cell will now take over by itself. What ends up happening is there are certain channels that are open that allow certain ions to flood cells, and other ions close to allow the ions to go out of the cell, okay? So most of the ions will flood inside the cell. And so if the outer portion of the cell is more positive, and those positive ions, well, in this case, sodium, are allowed to flood inside the cell, what that's going to do is going to make the cell more positive, okay? And so when it makes the cell more positive, it changes the charge, and the charge starts getting more and more and more positive as more and more and more of those sodium cells rush into the cell membranes. So these channels open up wide. Think of it like a floodgate, okay? And these positively charged sodium cells rush inside the membrane, making it more and more and more and more positive till it hits +30. Now, +30, that is where we say that the impulse has occurred. The electrical charge has occurred. Okay. And then once that happens, once that +30 is met, the electrical signal is sent to the next cell. It has fired, it has activated, it has done its job. Well, at this point, it's got to get back down to that resting state because it can't stay at this +30 forever. And so this enters what we refer to as the refractory period. Okay. So this refractory period here is in essence when all the sodium ions are kicked out of the cell, okay? And so those gates open up, okay? Those ions move out of the cells, it's the cell, and then we get more and more and more negative. Now, there is a period of time here, what we refer to as hyperpolarization, that the cell dips below its resting potential. Okay, and it may dip below as much as -90 millivolts, but then it very quickly will go up to the resting potential. During this period, we call this refractory period. I like to think of this as when you flush the toilet. If you flush the toilet and there's not enough water to fill the bowl yet, you can jiggle the handle as many times as you want. That toilet will not flush. It is in a refractory period. Most of the time, a cell that's in a refractory period cannot activate or fire again while it's in this period. Now, there are some rare occasions, there are some things that happen, but generally speaking, during this refractory period, that cell is done for that period of 0.5 millisecond. Now, during this so that's the whole process. Now, this period right here, that where the ions are moving across that axon membrane, this process from here to here is what we refer to as depolarization. So this is the depolarization, okay? And this is the hyperpolarization, okay? So what I'd like for you to do now is to just stop the video. I'm going to end the video now. I want you to watch this YouTube video because it's very good visual of how it works, because then you can see how the cell receives the stimulation from another cell and then how it has the electrical stimulation throughout the cell from the axon hillock all the way down to the terminal bouton. Once the cell has been activated, it's what we refer to as all or none. It either fires or it doesn't. There's no partial firing or maybe firing or kind of firing or pieces of firing. It either activates or it does not. So it's the law that the neurons either fire or they don't. So it's all or none. Okay. So go ahead and play that video so that you can get a good visual, and then move to the next chapter part.